okay? So what am I talking about? All I'm saying now is that the chemical composition of a hormone determines how it communicates with the cell, okay? Its chemical composition determines whether it's, its solubility, right? Whether it's going to, diffuse into the cell, whether it's going to dock on a receptor. Let's do some more stuff with that. So that's a general principle. Then you guys would need to learn a little bit more about the specifics, right? So we know that when hormones are released, they're going to be released, right, from the glands. They're going to target via, they're going to travel to a distant target in the body. So what are the examples maybe that you've learned about in Bio 11 is you probably learned about growth hormone. And growth hormone targeting, let's say, the long bones and stimulating proliferation and bone deposition so that your bones grow longer and you get taller. Maybe an example like that. Okay? So, what I'm referring to in my example about bones is the target. Okay? So hormones are going to travel in the blood to the target, and that's where the message will be communicated. So a growth hormone would talk to a bone. It would say, let's proliferate, let's lay down some bone matrix here, let's get growing. Right? That would be the talk. So everything that we learn about in the endocrine system is about that, right? It's about communication, right? Between the hormone and between the target. And it has an ultimate goal, right? So the ultimate goal of a growth hormone is growth, right? But hormones all have different goals. But they all have a common goal. And the common goal is to keep you alive, right? And to contribute to homeostasis, right? Because homeostasis is health, isn't it? That's the goal. So your dynamic system, you're changing all the time. Your nervous system and your endocrine system, their job is to pay attention to that flux, to all the changes that are taking place, and to respond. And to coordinate activities between all the systems in the body to get you as close to an even keel as possible. We call that homeostasis or health. Okay? So that's the big idea, right? So don't get mired in all the, every hormone you talk about, right? That's the end game. That's the goal. That's what it's going for. And in your brain, basically, what is it targeting? It's targeting the cells within that target, telling them what they should do, how they should behave. And in the case of the long bone, grow. All right. So now let's drill down into this a little bit, okay? These are specifics that you need to know, right? So anytime I put your attention on something, and I say that, and I stop, I point, and I elaborate, that's my signal to you, right, that I expect you to know this. I want to start here, okay? So what we're looking at here is a list of cellular activity, are we not? Yeah. So we're looking at all of the metabolic activity that takes place inside a cell. So the second example that I referred to, the stimulates the synthesis of enzymes or other proteins, refers specifically to the process of protein synthesis, doesn't it? It does. What are the two? And a lot of times I found it helpful when I was, in school to put things in the form of a question, question and answer. So what are the two steps so don't answer out loud yet, write it down. What are the two steps in protein synthesis? What are the two steps in protein synthesis? Where do they occur? So just write that down. Do you know the first step in protein synthesis? You're thinking of DNA replication. That's what you're thinking of. You're thinking about copying the DNA, and what I'm asking about is how do we extract the message that's in the DNA, right? Messages for making protein. How do we get that out? Okay, transcription and translation. Okay, let's write that down, transcription and translation. So transcription occurs at a specific location within the cell. So my question to you is, does transcription take place inside the nucleus or outside the nucleus?
Inside.
Inside, where the DNA is, correct? What is the end product of transcription? What is the result of transcription? Yes, RNA. What kind of RNA? mRNA. Okay? So messenger RNA. Messenger RNA is a DNA transcript, isn't it? An mRNA transcript is essentially a DNA transcript. It's a copy of the genes for the particular protein that you want to make. Everybody okay? Is anybody not really sure what I'm talking about? Because I'm going to stop. We're in the nucleus now, are we not? Step 2, translation. Translation takes place where? Ribosome. Is the ribosome inside the nucleus or outside? It's outside. Okay. So translation takes place at the ribosome, right? And basically, what is the result of translation? What do you get as a product? Protein. You get protein as a product. You get a polypeptide as a product. Okay, so let's tie this to what we're talking about. So what we're saying is that one of the primary activities of a hormone is that it has the effect of stimulating a target cell to do these things, right? So one of the things that hormonal stimulation will do is to cause the target cell to synthesize enzymes or proteins, right? So that's why we're pointing this out right now. That's the importance of it, okay? So that's one of the effects that a hormone has, and now we're talking about the specifics. What is one word used to describe, don't call it out yet, I'll let you know when. What is one word used to describe alter the plasma membrane permeability or the membrane potential by opening and closing ion channels? What is a word that we could use to describe that? What would you, or a bunch of words, one word or a few words? Okay, go ahead. Who's got something? Transduction, good. So what is transduction?
Transduction is the environment is changing how's the action of the potential. So for example, the heat, right, can change the surface of the membrane and the gates.
Okay, I'm going to add a word to what you're saying. So you are saying that, you gave an example of a stimulus. So you're saying it is the changing or transforming a stimulus into an action potential. Yes, that's one. What else? What is this referring to?
Junction.
Say it louder.
Junction.
Junctions? Are you saying junctions? What about them?
I forgot what it's called, but there's the one that's always open. That's the channel one, and then there's the one that's
Okay, so maybe you're talking about membrane gates. Okay, so when you move material into and out of a cell,
The transport.
all right, the word transport, right? Transport is the actual process, right? Right, it's the cellular process. So it alters, so it's affecting protein synthesis, hormone action on target cells are going to impact the transport mechanisms in the membrane. So we can take that descriptor, right, and we can look at it and say, what does this mean, right? It means that we're talking about the transport processes, whatever they may be, right? Whether they're a transduction event or not, right? Or whether they're whether it's gates opening and closing. But it influences the activity, the transport activity within the membrane, doesn't it? Yeah. So membrane's primary components, right? Don't say it out loud. The primary components of the plasma membrane are, write them down. The primary components of the plasma membrane are, don't say it out loud yet. What are they? Do we remember? Just think about it. Think. Don't let other people think for you. Always answer yourself. We all have different processing speeds. And some people take they just need another minute. So give them that minute to recall. Composition of the plasma membrane. So, in the back, let's hear from somebody in the back. Who can give me one plasma membrane component?
Phospholipids.
Phospholipids. What's another important membrane component? Go ahead. Louder. Proteins. Proteins have a lot of function in your membrane, don't they? Yeah? Okay. Now, there's a bunch. Let's see if you can come up with write down three functions of plasma membrane proteins. Those proteins are what are otherwise known as integral or peripheral proteins. You learned them? There's a bunch of functions. See if you can come up with three. Plasma membrane proteins function as
Here.
what?
I'm
Raise your
ready.
hand.
So this is it.
Raise your hand. Who's got one? Okay.
So this was
They
the first
function
one.
in transport as a channel. Can I say that? As a channel or gate?
And
Good.
then
Let's get another one. Who's got one? Okay, wait.
three
Let me see if I get something. That's
functions.
okay. That's good. We just want to see if we get somebody else. Guys, how about here? Anybody got one for me?
Membrane proteins.
Okay, thank you.
Mhm.
3.
She just gave that one, transport. That's good. That's all right. Transport. Come on, guys. I'm going to use yours. Junctions, cell joining, right? How about receptors? Yes?
3 what?
Yeah. Do plasma
Name three
membrane
functions
proteins
of
function
membrane
as receptors?
proteins.
Yes or no? Yes. Your Bio 11's coming back to get you. Your professor told you it would. I mean that in a nice way. I'm not the nice person. Trying to have some fun, right?
Are we still listing all functions?
Pardon?
Are we still listing all functions?
We
for
are
the
just
membrane
trying to get
proteins?
one more.
Structure.
How? I already stated that. We already
Right.
identified proteins as a structural component.
Cool.
Now what we're doing is we're identifying them as the functional aspect of that structural component.
Okay.
Wait. Come on, guys, one more. You can do it. Communication is receptors. We said transport. So we got receptors, transport, enzymes, joining. Oh, we already got three. Okay. I lost count. Okay. There's more than that, but that's good. Okay. Last stop on this slide. Here. Don't say it out loud. Stimulates mitosis. Write down the steps in mitosis for yourself. A lot of people are smiling and laughing. Like, yeah, right. Yeah, right. So I want you to think about the cell cycle. I want you to think about G1, SG2, and then mitosis. Right? And we know that mitosis is just part of the cell cycle. Right? So cells are either growing, synthesizing, proofreading, and checking your DNA, and then dividing, and then they go again and again and again. So mitosis. So there's an acronym for mitosis. IPMAT. IPMAT. Interphase, prophase. You finish it.
Metaphase, anaphase, telophase.
Metaphase, anaphase,
What about
telophase.
cytokinesis?
Cytokinesis is part of one of those
Yeah.
processes.
So instead of just words on a slide in a presentation, when you do this, what we're doing right now is you're enlivening the information. You're thinking about what it is and recalling. Not just like, oh, mitosis, I learned that, I kind of remember that. Really? Do you really remember it? You know? So when you're going, right, when you're learning this material, you need to stop, elaborate, example, explain before you move on. All right, let's move on from that. Okay, so let's make these distinctions and let's talk about the interaction of a hormone with its target based on its chemical composition. I'm going to say that again. Let's look at right? Let's look at the chemical composition of hormones so that we can appreciate how they interact with the target. This is what we've said so far. We have said we're going to put our hormones into two categories, right? Amino acid derived, lipid derived, or cholesterol derived. So what we're doing now is we're taking that information and we're building on it. And what we're saying is those amino acid derived are the water soluble type. One thing that we're going to have to incorporate into this conversation ultimately or eventually is how the hormones are traveling in the blood to get to the target. We're talking about the target right now, right? So we're going to have to talk about whether a hormone is water soluble or not, is going to determine whether it can be dissolved in the plasma or not. Here's what I'm saying to you. When a hormone is produced, it travels via the bloodstream to a target. Do you agree? Do you agree that the blood, connective tissue, blood, is the liquid portion is of the blood, what is known as plasma, is primarily composed of water? Okay. So in order for hormones to travel in the bloodstream, the manner in which they travel is dependent upon their solubility, whether they're water soluble or lipid soluble. So if they cannot be solubilized, if they cannot, right, they're not water soluble, they're going to need the help of a transport protein to be carried through the blood. So we'll come back to that, because I just leapt over something very important. So we're not, just hold that question for a second. Come back to the target, all right? So let's look at the mechanism. So all we really want to look at here is we want to look at how the hormone interacts with the target, okay? And we basically have these two propositions. So it's an either or proposition. If it's water soluble, okay, it's going to act on a plasma membrane receptor. We're going to see a picture of what that looks like in just a second, okay? It's not going to enter into the cell. But what it's going to do is it's going to initiate a second messenger pathway. So if the protein-derived water-soluble hormone could talk, it would come up on the cell, right? It would dock on a plasma membrane receptor, and it would communicate its message to the receptor. The receptor would transduce, that's what you were saying before, the message, right? It would communicate the message, right, into a second messenger system, a G protein system within the cell. And then what would happen next? Basically the message, right, would be to increase your enzyme activity, increase your protein synthesis, increase mitosis, decrease mitosis. That's the message, right? So all we're looking at is the pathway. How does the information get from the hormone to the interior of the cell to alter the cell's activity? Are you with me on that? I want you to stop and summarize what I just said. Write it down. You're gonna gauge how closely you were listening to what I just said. Summarize. Try to summarize what I just said, and then I'll do it again. What was the point that I just made? Okay, take a look up here. Let's contrast that with the other class of hormones, right? So these now are, right, the cholesterol-based ones, right, the lipid-based ones. The lipid soluble, they're going to diffuse into the cell and act here specifically with an intracellular receptor. So in the first example, you've got a membrane receptor. In this case, the second case, right, of the steroid hormones, the receptor is not on the surface of the cell. It's not on the plasma membrane. It's inside the cell. It's intracellularly located. activate it. And that's going to activate, right, specific gene sequences to increase the cell's activity. So you've got these two examples, yeah? Okay. Now I want you to, you just did like a little bit of a summary. I want you to go back now and add this idea to it. So you've got this either or. By summarizing things in your own words, right, you're making meaning for yourself, and that's what learning is. Okay, talk to the person you're sitting next to for just a second about that idea, about water soluble versus lipid soluble. Just talk to the person like, what is this? What are we supposed to know? Go ahead, talk to the person next to you, behind you, in front of you. Come on guys, don't be shy. If somebody's shy, invite them into the conversation. Yeah, you're by yourself. As I said, these guys back here.
I'm actually not sure.
Me neither.
I'm fully intending on re-listening to her.
Yeah, I thought I was. I didn't want to say it again, but I don't know either. I don't be like, oh my
I'm
god.
glad we're all on the same page.
Yeah, me too.
Slides, they're all on Blackboard.
All of them.
They're all on Blackboard.
So all she just went over was the syllabus, right? I had her before. I know how she went over it.
Well, she went over the before you came. She went over the syllabus, and then when you got here is when she started going over the slides.
I mean, I came on time for the slide part.
Yeah.
So it's the same book as we had like for Bio 11? It's the same book as
I
we
know the
had
lab
for
manual.
11?
Same book. I'm
Just
not really
chapters,
sure.
I think, 16.
I think for the most part it's the same book, but the notes that is posted on Blackboard is another professor's summaries of those chapters.
Who's your lab professor?
My lab? Man,
It's probably yours. There's only 2 labs in here.
Looking, yeah, I have
Yeah.
him.
He's not
I don't know. I saw him on Read My Professor. I'm like, I wanted her.
You
But
may or may not agree with me, but what you just did a whole lot of processing, okay? You processed the information, right? You didn't just kind of read it in a book or read it on a slide. You were thinking about it, weren't you? Weren't you trying to articulate and kind of, put it in your head, fit it in? It's foundational. You need to build on it. You're all future healthcare practitioners, aren't you? Yeah.
Hope so.
You want to develop a practice. Most of you are, not all of you. I understand that. You want to develop a practice where, how am I going to learn all this important information? How am I going to organize it so that I can build on it? Okay, let's come back to this. So there are animations posted to your Blackboard which you can watch after class, which will actually animate what I'm going to talk about. I'm not going to go into the specifics, nor on an examination am I going to ask you the specifics of this pathway. What do I expect? I expect that you know the different pathways, peptide-derived versus steroid-derived, meaning whether does the hormone interact with a plasma receptor or an intracellular receptor. I would expect that you would know that, but I don't expect you to know for this pathway of the second messenger pathway, every single detail. It's not necessary. This is not a cell biology class. We don't need to do that. But what I am going to say is if you would like, and if you're very interested, and I hope you are, you'll take a few minutes to watch the animation that's posted to your Blackboard, which will show you this pathway. Okay? So in a general way, let's just take a look at what that pathway is. So we've established, right, that the protein-derived or the amylin-derived hormones are going to dock on an intracellular receptor. So if you don't remember, this represents the phospholipid bilayer, right? And then this represents an enzyme, adenylate cyclase. This is a very large, right, transmembrane protein. And then we have, it's a protein that's functioning as an enzyme. We have another transmembrane protein here that's functioning as a receptor, right? So transmembrane proteins have multiple roles, right? They're super, super, super important. So don't don't stress about this, but let's just look at the pathway. So basically, what the idea is this. The idea is how is this message that's encapsulated within the hormone, which is basically a chemical message, which is going to alter the cell's activity, how does it get communicated to the cell? If it can't diffuse into the cell, it's going to basically dock on the surface of this transmembrane protein receptor, and it's going to basically activate a second messenger pathway using a whole series of cellular proteins called G proteins. So basically, we can see the communication with the signal being relayed via the second messenger pathway with the ultimate goal, right, of altering cell activity. That's as deep as we can go. Whereas, if we look at the second type, in this example, we're looking at the steroid-derived ones. These are the ones that are soluble, right? So they're going to dock on an intracellular receptor. And so what this is picturing is picturing the nucleus. This is picturing, as it says here, the receptor hormone complex. So here's the hormone, here's the receptor. They complex, they bind. When they bind, they migrate. They migrate through a nuclear pore. They dock on a specific gene that they're programmed to target, and then activate the protein synthesis sequence, and that's what we see happening here. Here is translation. We see the generation of an mRNA transcript, right? So what's happening is the hormone, right, is initiating protein synthesis. That's what this entire figure is showing us. Here's the product of transcription, an mRNA transcript. An mRNA transcript is simply a copy of the genetic sequence for the particular gene that we're targeting to make maybe another hormone or another protein or another enzyme, let's say. Once that is produced, that mRNA transcript, right? This is protein synthesis step 1. Step 2, the mRNA transcript it like leaves, we say it sneaks out of a nuclear pore. It will then, this represents a ribosome, this is a ribosome, this is a ribosome, this is the mRNA transcript. What ribosomes do is ribosomes read. Ribosomes read mRNA transcripts, and they translate the information on the transcript. You're making a transcript right now in your notebook, aren't you? You're taking the words that I'm saying, and you're transcribing them. Maybe you're transcribing them into your native language. Maybe it's not even the language I'm talking. Maybe it's another language, right? You might be doing that. So this is what the ribosome is translating what's on the transcript. And what's on the transcript, right, it's a whole series of A's, C's, T's, and G's. If I take 3 of those nucleotides, right, DNA is made up of nucleotides. If I take those 3 nucleotides, just like words in a sentence, they each mean something, right? So an mRNA transcript is made up of a series of 3-letter words called triplets, and each one of them stands for an amino acid. So the job of the ribosome is to read and to translate. So the ribosome reads the transcript and says, oh, I need tyrosine, I need arginine, I need leucine, I need, okay? And it assembles the polypeptide, one amino acid at a time. Do you see this beaded string here? That represents the protein or the newly synthesized polypeptide. I do expect you to know this. So translate, no, I'm so glad you said that because I said it in passing and you're asking a good clarifying question. So yeah, you are right. And you picked up on exactly what I was emphasizing. I'm going to re-emphasize it. Translating. So when a ribosome is reading an mRNA transcript, it's written in the language of nucleotides. If you don't know what a nucleotide is the basic subunit of DNA and RNA. It's what they're made up of, right? They're made up of a essential carbon, a carboxyl group, an amine group, like it's a unit, right? And now we translate that mRNA transcript, which is in the language of nucleotides, into the language of proteins. I have a question. Don't answer out loud. Are proteins made up of nucleotides or amino acids? Amino acids. That's what this step does, right? To your question, Good question. And you picked right up on that. That was my follow-up question. You asked it before. You asked the question before I did. That's good listening. Translation is translating from a nucleotide sequence to an amino acid sequence. You think that's going to be on your first test? Yeah, it is. Yeah. Any questions about the point that I just made that was not clear? Anybody? So this process that we just looked at is the process we just saw here. A steroid hormone stimulate this cell to produce a protein product through the process of protein synthesis. That's what we saw. That's what we're seeing here. We're seeing hormone action. We got a little bit more to do before we go. And oftentimes I like to just pause at this point because I know once I hit that 11:20, you guys are out, regardless of what I say. Which I get. We all have lives. We got things to do. So sometimes I like at this juncture, because I know I still have your attention, I'll make like important announcements and things that I want to say to you. My recommendation to you, and I told the students who are in my lab section this yesterday, if I were you, I would be working on the laboratory review sheet for the endocrine system. So your laboratory manuals, right, they are organized into exercises. Those are listed on your lab syllabus, the numbers, okay? The end of the lab activity, the protocol, the dissection, the wet lab, whatever we're doing, it culminates in a review sheet. That review sheet is a great tool for you to use for the endocrine system. It's going to pull in and organize all this information for you. So I highly recommend that you get started. And you can do that on your own and get started. You don't have to wait, okay? And that's going to be a great tool for your first test as well. So I recommend that you do that. Second thing I recommend that you do today is get onto Blackboard and look at what I have there, okay? And, consider watching the animations or at least some of the ones that are posted there. They're pretty good. We'll be continuing this endocrine lecture when I see you again on Monday. So here's our last point for today. It's actually two last points for today. Okay, let's look at these terms. So the number of receptors to a particular hormone, for a particular hormone is not fixed. It's variable. And it's variable based on the amount of circulating hormone. Okay? So what does that mean? It means that the number of receptors varies or changes based on the availability of the hormone. Okay? So cells are very sensitive, right? And responsive, unless you might have an endocrine disorder where that's not the case, but I'm talking about, right, the case where everything is functioning properly. So we use these terms upregulation and downregulation when we refer to the manner in which receptor numbers increase or decrease based on the availability of a hormone. Look at the statement. Upregulation, up specifically refers to the generation of more receptors when low levels of hormone are available. Kind of makes sense, doesn't it? If there's not a lot of hormone available and you up the number of receptors, then you're maximizing the functionality of the cell, right? If the hormone receptor number stayed the same and you had low levels, you would have low cell function, wouldn't you? Let that marinate, right? The converse is true also. When levels are super high, look at the statement, targets lose receptors in response to high levels. So they become less, the cell becomes essentially less sensitive. Less receptors, less sensitive, more receptors, more sensitive. That's an important term. And then finally, our last point for today. So it's a question, right? So the question would be, what controls the release of hormones? What kind of stimuli result in hormone production and hormone secretion? There's a bunch in the list at the bottom, right? Humoral, neural, hormonal. So the term humoral, we'll elaborate in just a second. When you see humoral, you should think of blood plasma levels. Right, so humoral specifically refers to body fluid, and specifically is really referring to the blood or plasma Neural is what it sounds like, right? Nervous stimulation, and so is hormonal, right? So we might have a hormone acting on another hormone. All right, first example, one that we're already familiar with, right? Parathyroid hormone and calcium homeostasis. By the end of the next lecture, we're going to develop our understanding of calcium homeostasis because it's not as simple as this, okay? Your calcium homeostasis is not just predicated on the amount of parathyroid hormone you have. It's going to be more complicated than that. Okay, so this is also a Bio 11 sort of recall, right? So we're bringing it back, and we're going to be learning more about calcium homeostasis, especially with respect to the heart and to other systems of the body, right? But you learned about calcium homeostasis and parathyroid hormone. But you learned specifically that parathyroid hormone targets a specific type of cell and changes its activity. What is the name of that cell? Osteoclast, right? Osteo means bone, right down, yeah. Right, means bone, and clast means digestion. So bone digesting cell. And when osteoclasts digest bone matrix, they liberate the calcium that's stored there, right? That's what your bones are doing for you. They're your calcium warehouses, right? That's where the bone is. It's not about the free circulating calcium in your body, right? It's really about where the calcium is deposited in the body, and then it becomes available. Okay, so a humoral stimulus means that when calcium levels drop low, right? Low levels of calcium in the blood, right, is a type of humoral stimulus. It's a stimulus which will then cause the release of parathyroid hormone to correct that calcium deficiency. So the question is, why was the parathyroid hormone released? Was the stimulation humoral, neural, or hormonal? What caused it? It was humoral. The stimulus was humoral. It was based on blood levels of calcium. Was it neural? No. Was it hormonal? No. Right? It was based on the levels of calcium that were in the blood. And then neural stimulus, cross-section of the spine, and then here we have a cross-section of the adrenal gland, which we haven't addressed yet. And what we can see here is some autonomic innervation of the medulla of the adrenal gland. So in some cases, right, hormones are going to be released based on neural stimulation, as in this example. And then finally, hormonal stimulation. So this is something that, again, more to follow next week on Tuesday. What we have pictured here is the both of the lobes of the pituitary gland. And what we can see is that the anterior lobe of the pituitary gland is producing various, these little dots represent hormones, and they are targeting specific glands. The thyroid, the adrenal, and the testes, and stimulating them to produce their secretion. So it's hormone on hormone, right? Pituitary to gland, it's a hormone, and then the gland produces its secretion. So we would say when the thyroid gland produces its secretion, T3, T4, collectively known as thyroxine, what causes that to happen? Is it nervous stimulation? Nope. Is it humoral? No. Hormonal. Okay?